Hello, my name is Samara Singh. I'm a PhD graduate student at the University of Miami working in Dr. Nipun Merchant's lab. I'd like to thank the APA for inviting me to share my abstract today, and I will be sharing my work on targeting IL-1α production through p38 MAP kinase inhibition in KRAS mutant pancreatic cancer. Here are our disclosures. Previous studies in our lab have focused on interleukin-1 alpha, specifically as a key mediator of tumor progression and resistance to therapy in pancreatic cancer. We've shown that it's overexpressed and correlates with worse overall survival, and blockade of IL-1 improves survival outcome in mice by remodeling the tumor microenvironment. Interestingly, others have shown that IL-1 alpha is a downstream product of activating KRAS mutations in PDAC tumor cells. However, the mechanisms mediating IL-1 alpha production downstream of KRAS remain unknown. The purpose of this study was to identify targetable kinase pathways downstream of KRAS that are involved in IL-1 alpha production. To do this, we did a targeted drug screening using inhibitors of major nodes downstream of KRAS and looked at IL-1 alpha during transcription and response. Interestingly, with p38 MAP kinase inhibition, we saw most significant reduction of IL-1 alpha expression reproducibly in multiple PDAC tumor cell lines, implicating its importance in IL-1 alpha regulation. In line with these findings, we looked into activated p38 MAP kinase levels in human PDAC tissues and noted a substantial increase of phosphorylated p38 in tumors. Interestingly, it appeared to be increased in the ductal compartment, supporting our hypothesis that this pathway is KRAS-driven. To further explore p38 MAP kinase activity in IL-1 alpha's regulation, we inhibited p38 using Pexmetinib, an orally bioavailable inhibitor currently in clinical trials for myelodysplastic syndrome. Treatment of various PDAC tumor cell lines with Pexmetinib demonstrated potent dose and time-dependent reduction in p38 phosphorylation, confirming targeting inhibition of the p38 pathway. And when we examined IL-1 alpha gene expression in these cell lines, we saw a significant reduction with p38 inhibition. Interestingly, we saw that Pexmetinib may have direct anti-tumoral effects, suggesting inhibiting p38 may also have a potential therapeutic benefit by reducing tumor growth. Others have shown that IL-1 is a potent activator of p38 signaling, and to investigate this, we treated tumor cells with exogenous IL-1 and saw p38 MAP kinase is activated with IL-1, suggesting that IL-1 alpha downstream of KRAS may sustain its own production through a feed-forward mechanism involving p38 MAP kinase, and we plan to explore this in future studies. In conclusion, IL-1 alpha is overexpressed in PDAC. p38 inhibition reduces IL-1 alpha gene expression in PDAC tumor cells. IL-1 activates p38, suggesting a feed-forward loop in KRAS mutant PDAC tumors, and p38 may be a potential target to reduce inflammatory signaling and improve outcomes in PDAC. I'd like to thank everyone in the lab for helping me greatly with this project.